Hello, and welcome to our podcast, Resistance Rising. Can we outpace AMR and MDR? In this podcast, we explore one of the most pressing yet often underestimated threats to global health, antimicrobial resistance or AMR. As antibiotics and other antimicrobial agents lose their efficacy, we are inching closer to what some call the post antibiotic era. Multidrug resistant organisms, once confined to hospitals, are now becoming alarmingly common in communities, livestock, and even the environment. But resistance is not an abstract threat, it is rooted in biology, policy, behavior, and economics. And understanding how microbes evolve and spread resistance is critical to fighting back. Through a scientific lens, we will explore how resistance emerges, how it's tracked, and what global systems are doing or failing to do in response. Let's begin. In segment 1, we will discuss the science of AMR and MDR, mechanisms of resistance. At its core, antimicrobial resistance is an evolutionary process. Microbes, bacteria, fungi, viruses, and parasites develop strategies to survive the drugs designed to kill them. These strategies are not random, they are driven by selective pressure. When antibiotics are overused or misused, resistant strains survive and multiply, while susceptible ones die off. Over time, this leads to populations that can evade multiple drugs, resulting in what we call multidrug resistant or MDR organisms. There are several key mechanisms by which resistance occurs. The first is gene mutation, a spontaneous change in the microbial DNA that alters the drug's target or function. For example, a single point mutation might prevent an antibiotic from binding to a bacterial enzyme, rendering the treatment ineffective. Another mechanism is horizontal gene transfer, where microbes exchange resistance genes through plasmids, transposons, or bacteriophages, an evolutionary shortcut that spreads resistance rapidly between species. Efflux pumps are another common strategy. These are protein structures that actively expel antibiotics out of the microbial cell before they can exert their effect. Meanwhile, biofilm formation creates a protective matrix that shields entire colonies from antibiotics, making them 100 to 1,000 times more resistant than free floating cells. Understanding these mechanisms is crucial not only for developing new drugs but also for designing diagnostics that can detect resistance early. With resistance evolving faster than our drug pipeline, the science behind these microscopic defenses holds the key to survival. Thank you.
Segment 2, Global Drivers of Resistance from Overuse to Misuse. Antimicrobial resistance does not occur in a vacuum. It is driven by human decision, medical, agriculture, economic, and political. Perhaps the most prominent driver is the overuse of antibiotics in human health. In many parts of the world, antibiotics are still prescribed for viral infection where they offer no benefit. In some regions, they are available over the counter without prescription, fueling inappropriate use. But misuse isn't just a clinical issue. Agriculture accounts for more than half of global antibiotic consumption, particularly in livestock farming, where antibiotics are used for growth promotion and disease prevention in crowded, unsanitary conditions. These practices create reservoir of resistant bacteria that can jump to human through food, water, and the environment. Then there is a lack of access to diagnostics. In settings where lab facilities are limited, clinicians often prescribe broad spectrum antibiotics just in case, inadvertently encouraging resistance. Without diagnostic tool to identify the pathogen and its resistance profile, targeted treatment is impossible. Another silent contributor is inequity, where the overuse of antibiotics in high income countries coexists with lack of access in lower income ones. This imbalance allows resistant infection to flourish in the shadows, often goes undetected until they spread. To address AMRS globally, we must not only innovate in laboratories but also intervene in policies, education, and behavior across sectors, from pharmacies and clinics to farms and food systems.
As resistance evolves and spreads, the need for rapid detection and global surveillance has become critical. Traditional culture based methods are no longer sufficient, they're too slow, too narrow, and often miss emerging resistance genes. Instead, we now rely on genomic tools that can capture a pathogen's entire resistance repertoire in a matter of hours. Whole genome sequencing, or WGS, allows scientists to identify not only the species but also the specific mutations or resistance genes it carries. During outbreaks, WGS can trace the origin and transmission pathways of resistant strains, enabling faster containment. Platforms like Oxford Nanopore and Illumina have made sequencing portable, affordable, and scalable. To interpret this deluge of genomic data, we need curated databases. Tools like the Comprehensive Antibiotic Resistant Database or CARD and ResFinder analyze sequences and flag known resistance genes. These platforms are essential for clinical labs, public health agencies, and researchers working in real time pathogen surveillance. Moreover, global initiatives like the WHO's GLASS, which is Global Antimicrobial Resistance Surveillance System, aim to harmonize resistance tracking across borders. However, these efforts are still fragmented with large data gaps, particularly in low income countries where infrastructure and internet connectivity limit participation. Genomic surveillance offers a window into the microbial world, allowing us to anticipate resistance before it becomes untreatable. But to make it truly effective, data sharing, interoperability, and equitable access must become central pillars of global health policies.
As resistance renders our current antibiotics ineffective, we face an urgent need for new therapies. But antibiotic discovery has long been in crisis. High developmental cost, low returns, and scientific hurdles have driven most pharmaceutical companies away from the antibiotic pipeline. Fortunately, new strategies are also emerging. Artificial intelligence and machine learning are now being used to scan vast chemical libraries and identify novel antibiotic candidates. One landmark example is the discovery of helicin, an AI predicted molecule with broad spectrum activity, including against multi-drug resistant pathogens. Natural products are also being revisited, especially from unexplored sources like soil microbes, deep sea organisms, and plant secondary metabolites. Meanwhile, bacteriophages, viruses that infect bacteria, are gaining renewed attention as precision tools that can target specific strains without disrupting the microbiome. In parallel, researchers are exploring drug repurposing, identifying existing non antibiotic drugs with antimicrobial properties. This approach dramatically reduces development time and cost as safety profiles are being established. These innovations represent hope, but they are also required sustained funding, regulatory support, and global access strategies. Without equitable distribution, the new solutions could become tomorrow's luxury drugs, deepening the divide between those who can treat resistant infections and those who cannot.
Now, let's talk a little bit about infection control and stewardship in hospitals. Hospitals are ground zero for antimicrobial resistance. The very settings designed to heal can become breeding grounds for resistant organisms, particularly in intensive care units, surgical wards, and neonatal units. But they are also where meaningful change can be enacted. Infection prevention and control measures, IPC measures such as hand hygiene, equipment sterilization, and environmental cleaning, form the frontline defense. Hospitals with robust IPC programs consistently report lower rates of healthcare associated infections, including those caused by MDR organisms. Equally important is antimicrobial stewardship, the coordinated effort to optimize antibiotic use. Successful programs include clear prescribing guidelines, real time feedback for clinicians, and pharmacist led review teams. In some countries, hospital accreditation now depends on active stewardship protocols. Technology plays a growing role. Clinical decision support systems, electronic prescribing platforms, and integrated diagnostics can help prevent unnecessary prescriptions and flag high risk patients. However, implementation remains uneven. Many hospitals, especially in under resourced settings, lack the training, infrastructure, or institutional mandate for stewardship. Bridging these gaps is not only about funding, it's about leadership, accountability, and a culture of safety that places responsible antibiotic use at its core.
Antimicrobial resistance is not confined to hospitals or human health. It circulates across humans, animals, and the environment, an interconnected web that demands a unified response. This is the essence of the One Health approach. In livestock farming, antibiotics are used not only to treat infections but also to promote growth and prevent disease in intensive rearing systems. These practices create reservoirs of resistance genes that can spread to humans through meat, water, and farm workers. Surveillance has detected resistant E. coli, Salmonella, and Campylobacter in meat products worldwide. The environment serves as a vast, largely unmonitored reservoir. Wastewater from hospitals, pharmaceutical factories, and farms often carries antibiotic residues and resistant bacteria into rivers, soil, and groundwater. In some urban slums, open defecation and informal waste dumping create hotbeds of resistant pathogens. Integrating veterinary surveillance, environmental monitoring, and public health data is essential to understanding how resistance flows through ecosystems. The FAO, WHO, and OIE have all emphasized One Health in their AMR strategies, but practical integration remains limited. We cannot address AMR in humans without addressing the agricultural and environmental dimensions. A siloed approach will only shift the burden, never eliminate it. One Health must move from rhetoric to reality if we are to contain the next generation of superbugs.
Segment 9: Public awareness, education, and behavior change. No global strategy against AMR can succeed without the full engagement of the public. After all, every time an antibiotic is used, rightly or wrongly, selection pressure is applied. Patients, caregivers, farmers, and prescribers all shape the resistance landscape through their everyday decisions. Unfortunately, AMR is still poorly understood by the general public. Misconceptions abound that antibiotics cure viral infections or that stronger antibiotics are always better. In many countries, people can access antibiotics without a prescription, often guided by informal networks or incomplete advice. Education efforts must therefore extend beyond the healthcare setting. School curricula, public service announcements, and social media campaigns can raise AMR literacy and foster more judicious use. Behavior change interventions like peer influence, visual nudges, and reminders have shown promise in shifting prescription patterns among clinicians. Healthcare providers do need ongoing training. Many operate under pressure from patients or time constraints, defaulting to antibiotics to avoid risk. Empowering them with diagnostic tools, decision support systems, and clear treatment guidelines can improve confidence in non antibiotic pathways. Ultimately, changing behavior is about changing culture from one that views antibiotics as a default safety net to one that recognizes them as a finite shared resource. Without this shift, policy and science alone will not be enough.